In this video, we will discuss the introduction to metabolism. Metabolic pathways are a series of consecutive enzymatic reactions wherein the reactants or substrates produce specific products by utilizing or by releasing free energy. They are either catabolic or degradative pathways and anabolic or biosynthetic pathways. Metabolic pathways are usually irreversible, that is, highly exergonic reactions. Most metabolic pathways are controlled by regulating the enzymes catalyzing their first committed step. Metabolic pathways in eukaryotic cells occur in specific cellular locations. For example, ATP and acetyl CoA are generated in mitochondria, but their function is seen in the cytosol. Biochemical reactions involved in metabolism can be broadly classified into oxidation reduction reactions, ligation requiring ATP, isomerization, elimination and rearrangement reactions, group transfer reactions, and hydrolytic reactions. Let us discuss few of the examples for in each category. Firstly, oxidation reactions such as succinate getting converted to fumarate. Here, both the hydrogens are removed and they are taken by FAD and getting reduced to FADH2, and a double bond is formed between C2 and C3 carbons. And the next example involves malate getting converted to oxaloacetate. Malate's second carbon has an OH group and a H, and these two hydrogens are useful in the reduction of NAD getting converted to NADH plus H plus. And this malate gets converted to oxaloacetate by forming a carbonyl group. Next comes the ligation reactions. Herein, the pyruvate plus carbon dioxide helps in the formation of oxaloacetate, and ATP is hydrolyzed to ADP plus PI. Next comes the isomerization reactions. In this, citrate gets converted to isocitrate. Isocitrate is the isomer of citrate wherein the second carbon's hydroxyl group is shifted to the third carbon. And next comes the group transfer reactions wherein glucose takes a phosphate group from the ATP and forms glucose 6 phosphate. Next comes the hydrolytic reactions wherein a bigger peptide is broken down into smaller peptides by taking in a water molecule. In contrary to this is the condensation reactions wherein a small peptides combine to form a bigger peptide by releasing water molecule. Next comes the addition of functional groups. When a functional group is added or removed from a compound, there is a possibility of double bond formation. This is the basic schematic representation of metabolism, wherein the complex molecules like proteins, carbohydrates, lipids gets converted into their corresponding amino acids, glucose and fatty acid and glycerols. These in turn gets converted to pyruvate and acetyl CoA, and this acetyl CoA is oxidized to carbon dioxide through TCA cycle, and the NAD plus and FAD molecules gets reduced to NADH and FADH2. Reoxidation of these NADH and FADH2 through oxidative phosphorylation releases ATP and water molecules. Complex molecules like proteins, carbohydrates, lipids getting converted into their corresponding monomeric units to amino acids, glucose, fatty acids and glycerols involve a wide range of biosynthetic as well as degradative pathways. We will see many of these pathways in the coming videos. The chemical basis of these reactions would be either nucleophilic or electrophilic. Electron-rich compounds are nucleophiles like ammonia, NH3+, OH-, imidazole, sulfhydryl, etc. And electron-deficient compounds like electrophiles are called electrophiles. These involve H plus ions, metal ions, carbonyl carbon, and imines, and many others. Taking the example of ATP hydrolysis, um, ATP hydrolysis can take place in two possible ways, that is ATP getting converted to ADP plus PI or to ADP plus glucose 6 phosphate, and ATP can also be hydrolyzed devoid of any enzymatic action. Choosing between one among these depend on the enzymes involved in the catalysis and its thermodynamic favorability or acceptance. This is the reason why biological reactions are highly specific. Thank you.